Hi, my name is Dr. Georgia Clarke and I'm from the Vagal Afferent Research Group, which is situated within the School of Biomedicine at the University of Adelaide in Australia. My lab's primary research goal is to understand the role of gastric vagal afferent signalling in the regulation of food intake in both health and disease. Within the stomach, gastric vagal afferents primarily respond to mechanical stimuli, or to stretch. This occurs as the stomach fills, activating tension-sensitive vagal afferent neurons, which then send satiety signals to the brain. Gastric vagal afferents are highly plastic, allowing for food intake to adapt rapidly to changing metabolic demands. Previously, our group has shown that from mid-pregnancy onwards, and in mice fed a high-fat diet, their stomach's response to stretch is reduced, and there is an increase in food intake. We were then really interested in the combination of obesity and pregnancy, since it is becoming increasingly common in society. We therefore randomized mice to either receive a standard laboratory diet or a high-fat, high-sugar diet for 12 weeks. Then they were randomized to either pregnancy or non-pregnant groups. Gastric vagal afferent activity was measured at late pregnancy. This was the focus of our recently published article in the Journal of Physiology. So we found that in standard laboratory diet mice, gastric vagal afferent responses to stretch were lower in pregnant compared to non-pregnant mice, which is in line with increases in total food intake and meal size. We also report higher growth hormone receptor mRNA expression in the nodose ganglia in pregnancy, possibly accounting for these lower gastric vagal afferent responses in pregnant standard laboratory diet mice. Furthermore, gastric vagal afferent responses to stretch were selectively attenuated in non-pregnant high-fat, high-sugar diet mice compared to standard laboratory diet non-pregnant mice, and this is in line with changes in food intake. And lastly, despite higher food intake, there were no further reductions in the response to stretch in pregnant high-fat, high-sugar diet mice compared to non-pregnant high-fat, high-sugar diet mice. Therefore, future studies are required to increase our understanding of food intake regulation across pregnancy. Overall, this research is important given that obese women are entering pregnancy at an increased risk of pregnancy complications. Therefore, strategies to reduce energy intake or enable adherence to the nutritional guidelines are clinically important. Thank you for taking the time to watch this video. The full paper was published in March 2025, and if you have any questions, please contact the corresponding author, Professor Amanda Page. Thank you.